Hello and welcome to this session on PAT and digital quality management in the life science industry. Let me first take you back to the breakfast table this morning, and I hope you did enjoy a soft-boiled egg on your breakfast plate. However, I also hope that you have not been thinking about how long this egg had to be boiled for achieving this result. Because if you would ask this question to 10 different people, then you probably would get 10 different answers, varying from 2 and 2.5 minutes to 5 or 6 minutes and everything in between. And you can't hardly say that any of these answers is right or wrong, because even with a simple process like boiling an egg, there is variability that comes into such a process. Think simply about the fact that the chickens that have been producing this egg have maybe been running around in the free open space, or could also have been chickens that have been squeezed with 1,000 chickens in a square meter. For sure, those chickens will generate eggs with a different quality level than the first ones. And that is what you simply don't know when you're cooking that egg or preparing that egg for your breakfast. So wouldn't it be nice if we would have a tool available that can measure inside the egg and give us information about the chemical and or physical condition of the yolk inside the egg while we are boiling it, so that that real-time information can help us to decide to take out each individual egg out of the boiling water at the appropriate time to have the perfectly soft-boiled egg on our breakfast plate. Unfortunately for cooks, such a tool does not exist as far as I'm aware. But fortunately for the pharmaceutical industry, there is technology that helps to get that information for the drug product manufacturers. Let's have a view at how it is today. We have typically a fixed process, fixed because of the fact that it is validated and therefore not to be touched. Nevertheless, variability still comes in through raw materials, through process conditions, through operators that are interfering. Anyway, that variability might have an effect on the end product quality. Now, if we are not allowed to touch the process because it is validated, variability that comes in will also come out. The only question is it acceptable or not? And that is what we are then typically checking at the end of the line by taking some samples and have them analyzed in the lab. That approach, typically called the quality by inspection approach, is what we want to get rid of by moving into the direction of quality by design-based approach. Quality by design-based approach means that we are going to design our processes such during the process development stage that we have a better understanding on what's going on inside the process, and that we also know that in case variability enters into the process, and when we detect that variability, that we also know how to react upon that variability and make sure that the effect or the potential effect of the variability on the end product quality of what we are making, that can be minimized or even be reduced to zero, so that we have a consistent output of our processes and that we are producing right first time without any bad product generations. So let's look at it in a more practical way, and let's have a look at the batch process in pharma. Typically, there will be a control system on top of that, making sure that the process is managed based on the traditional process control sensors, like temperature, pressure, speed, etc., etc. And then we are taking samples to the lab and have them analyzed. That is the quality by inspection approach that we want to get rid of, and we want to move into the direction of a quality by design-based approach. How does this look like in practice? Well, let's take an example of a fermentation process here. And typically, such a process unit will have a control system on top that is controlling the manufacturing process in such a unit based on traditional process sensors like temperature, pressure, stirring speed. What we would like to do now is to introduce also additional data sources that gives us information about the product itself inside the vessel. And that can be done by introducing PAT sensors in such a unit. Now, those PAT analyzers are typically spectral data sources like Raman, near-infrared, etc. And unfortunately, the closed-loop control systems in place are not designed from an architectural point of view to cope with spectral data sources. They are very performant and strong in treating single-value sensor outputs, but not spectral data sources. And that's where Siemens offers you the possibility to already introduce those PAT analyzers and integrate them into your existing infrastructure without drastically changing or impacting that infrastructure. As you can see on the screen, SciPat's first function will be to make sure that we are capturing those spectral data sources in real time. That is exactly where SciPat is designed for, so that is no problem. And it can even combine it with other data coming from other data sources at the same time. SciPat will then make sure that all these data are put into a prediction model that gives you, at any moment in time, a view on the predicted quality of what you're making in your manufacturing unit at that moment. So that you already know upfront what the end product quality will look like at any time. Having that view on CQAs in real time is the first step. In case the CQAs tend to go out of spec, we need to intervene. In that case, we need to close the loop and come to an advanced process control strategy, which will help us to keep the CQAs under control by managing new set points for those process parameters that have a major impact on the quality attributes that we want to control. If we manage in the end to keep all the CQAs for that process and for that product under control during the execution of that fermentation time, which can take many different weeks, then we are sure that the product that leaves the vessel will be of right quality and we can release it to the market without any additional testing in the laboratory, which can often be very costly and time-consuming. This picture gives you then an overview of what SciPath is integrating to achieve this goal in the end. And this was now explained for a batch process. Now let's have a look at the continuous line. What does change? Well, as you can see on the screen, not a lot, except for the process itself. But you can imagine that in a continuous line where everything is linked to each other, that an integrated approach of PAT integration is even more essential and more key than on a batch process. Siemens has been involved from the very early days of continuous manufacturing. Even one of the first products that has been approved by the FDA based on continuous manufacturing with a PAT-based control release strategy on it has been realized with the involvement of Siemens with PCS7 and SciPath on it. And in the meantime, we have done many different projects on continuous manufacturing as well. But continuous manufacturing might be a big driver for PAT. It's not definitely the only one. There's for sure still a reason to apply PAT on batch processes as well, as it is often the case, for instance, also in the large molecules area, where we see here the example of a fermenter, where a Raman instrument is being used as input to feed data into a glucose concentration model that is predicting the glucose and lactate concentration for that unit. And that is then being used, that input is used as a basis to calculate the amount of glucose that needs to be added to the fermenter to optimize the yield of the process. And this is really done in a real-time closed-loop control system. But these are just two simple examples, high level, to indicate that both on batch as well as on continuous, PAT can be applied. In pharma today, PAT is applied in all areas, both R&D as well as manufacturing, both formulated products as well as drug substances, small molecules as well as large molecules, batch processes as well as continuous processes. And in the meantime, also outside the pharma, there is a need for PAT, and there's more and more requests to also apply PAT on other processes than the pharmaceutical ones today. Today, PAT may indeed be applied in different areas of the pharmaceutical world. It has always been considered as a very innovative technique for the pharma. And it still is. It still is part of a lot of initiatives that our pharmaceutical customers are taking to investigate the art of the possible and to see what the factory of the future can bring for their operators and also for the efficiency of their processes. One of those examples is, of course, as already mentioned, the continuous manufacturing technique. And maybe we already consider it today as a given in the pharma. We should not forget that this is already something that has been applied in other branches for many, many decades. And only for the last 10 to 15 years has been introduced into the pharmaceutical world. Today, this is really exploding, and that's also what we have noticed when we have been organizing this event almost 2 years ago now, together with our partners, KEA and Perceptive Engineering, and where we have given a platform to our existing customers to tell their experience, to share their experiences on continuous manufacturing, PAT, and advanced process control. And there you see again, continuous manufacturing is linked to PAT, and PAT is combined with advanced process control to again bring some innovation into the approach that pharmaceutical manufacturing processes are being managed. But that's not the only example. We have also more recently been involved in a digital twin initiative from one of our key customers, GSK Vaccines, in the vaccine production process, where indeed PAT again played an important role as part of the complete setup. The special thing here is, and that's also where the strength of Siemens comes in, is that we have combined PAT and SciPath with other technologies that Siemens has been investing in, and that we have been combining those in technologies to create a complete digital process twin for GSK. Here you see an overview of the unit that we have been working upon together with GSK Vaccines. As you can see in the middle, we have been using computational fluid dynamics, or CFD technology, to switch the traditional big batch vessel into a very small, miniaturized, size of a smartphone, continuous unit, where we can continuously fill in the liquids on the one hand side and take out the desired liquid combination at the other side. As this is a continuous process now, we are also monitoring the quality in real time by introducing and integrating an NIR measurement tool via SciPath. And the third thing that Siemens has achieved here is as well to combine these real-time measurements of SciPath with a MPC control unit so that we have an even better control over the complete process by model predictive control applications. On top of that, machine learning technology is even helping to continuously improve the prediction quality of those MPC units. And with this, I'd like to conclude this presentation. I hope I've given you an overview of what Siemens can offer to help you with your digital quality management strategy today and in the future. First of all, we can help you with a real-time PAT integration into your existing processes and of course also in your new processes. Second thing is, and that has not been mentioned in this presentation even, is the fact that we can also help you with the creation of your more science-based mechanistic models of your processes through our GPROMS platform that we have acquired 2 years ago. And the third thing is that we can combine all these solutions to create a full digital twin of all your processes as well. Siemens has clearly all the solutions and competences in place to help you realize the complete path to your digital quality management strategy already today. Thanks for your attention, and I hope to hear from you soon.